The NanoAssemblr Ignite is a powerful tool for advancing your nanomedicine development. We took our NxGen technology, which was designed for GMP production, scaled it down, and designed a system around it to enable scientists of any skill level to explore the nanomedicine design space. In this demo, we'll be making enough lipid nanoparticles to encapsulate all the mRNA needed to meet the dosing described in the Gale 2012 paper. Based on the N/P ratios used, this can be achieved in a single 5 ml Ignite formulation. The Ignite's interface puts key parameters that define the conditions of particle formation, such as the total flow rate and the flow rate ratio, literally at your fingertips. It ensures that conditions are highly reproducible, run after run, regardless of who's using it. With Ignite, NxGen has been scaled down and engineered into a format appropriate for preclinical development work. Fluid input is handled through syringes which attach by Luer lock directly to the Ignite cartridge. Here, RNA in an acidic buffer is loaded in a syringe. Then our Genvoy ionizable lipid mix dissolved in ethanol is loaded in another. The Ignite will inject these into the NxGen mixer under the conditions specified to control the nanoprecipitation reaction. Because the same NxGen technology is available across the NanoAssemblr platform, users are able to work with and start developing a process representative of the eventual GMP process. So you're not only making formulations for preclinical studies, you're also getting a head start developing that final process at the earliest stages. The ease of use of the Ignite addresses another appreciable challenge faced by the field, namely assembling or training a workforce skilled in the art of making LNPs. The Ignite lowers the barriers to successfully formulating nanomedicines and reduces human error. This means you don't need to spend years in the lab perfecting a technique. Instead, new users can get up to speed very quickly, parameters can be transferred between users or to other departments or between collaborators, so researchers can concentrate their efforts on the multitude of other activities that are crucial to developing a vaccine or other nanomedicine. At the heart of the Ignite is this NxGen microfluidic mixer. We created NxGen to address the challenges of clinical and commercial scale manufacturing. On a GMP system, a single mixer throughput of over 200 milliliters per minute is possible, while maintaining the control and reproducibility that is the hallmark of NanoAssemblr non-turbulent mixing technology. The fluid path is completely disposable, mimicking what you would do in the GMP environment. This also reduces the risk of cross-contamination and eliminates the priming and cleaning steps, which greatly streamlines the workflow. Here you can see just how fast Ignite produces this formulation. You'll also see Ignite automatically divert a small portion of the formulation at the head and tail of the process to ensure only the best quality particles produced under steady-state conditions are collected. And here you have a 5 ml formulation which encapsulates more than enough RNA to meet all the in vivo dosing requirements of the paper. You can make formulations between 1 and 15 ml pre-dilution, which is ideal for exploring the formulation design space and evaluating formulations in a range of in vitro and in vivo models. The next step for this formulation is to prepare the particles for dosing. This involves particle purification by either centrifugal or tangential flow filtration, and finally sterile filtration. The ease of use, speed, control, reproducibility, and scalability make the Ignite a tremendously powerful preclinical development system.